everybody. Welcome to SDSF Live. Please let us know where you are logging on from. I'm Nicole Shen from Texas, and I have Michelle from Michigan with us. SDS Live is brought to you from the Shwachman-Diamond Syndrome Foundation, providing support to families around the world. We are currently live from Facebook and YouTube. You may comment from both platforms, and we'll see it on our feed. The show is public. Please do not ask or share personal information that you are not comfortable sharing. These shows are served as a library of topics on YouTube for families to access later, where it could also be translated to other languages. Hi, Michelle. How are we doing today?
Hi, everybody. We're doing fantastic. It's sunny here. It's above 50 degrees, and I will take it. And the leaves are turning. I don't know if Nicole said this, but when you do log on, let us know where you're logging on from. I'm Michelle Grinnell. I'm from Michigan, and I'm happy to be here. We have a pretty exciting super packed show today. So I want to get right into it. I want to show you guys the giveaway that you can earn or win today by interacting with the show in any way, liking, commenting, sharing, inviting people, starting a watch party. And I'm pretty excited about this. Nicole and I tried to figure out some cool things to get you guys. I don't know if you can quite see in the picture, but in the picture, there's a glass coffee cup that has the molecule of caffeine on it, and I really think it's funny. And then there's also a science kit for kids, because we're talking about some very important research today. So, Nicole, I'm so excited. I think I want to jump in and tell where people are logging in from. I see there's Missouri, London, New York, England, another UK, London, Alberta, Canada, Maine, Michigan, represent. I love Michigan. Ireland, Texas, Tennessee. Oh, look there's Dr. McCaslin, who is our pulmonary doctor on our board of medical directors at SDSF, and he's logging in from Florida and warm and sandy and full of alligators. Scotland, Cleveland. There's Dr. Corey. Such a packed show today. Such a packed show. Look at all of these. Oh, I love it. More England, more England. I love that we have everyone here. I think it's really going to help pull in the vision that we want to show with all in.
Yep, definitely. Thank you, everyone logging in, and thank you, everyone, all the doctors joining. That's really exciting for us.
Yes. I love a lot of these doctors too are ones that we've had in the past on our show. And so it's kind of interesting to see them on the other side in the comment section.
Absolutely.
It's interesting. Very interesting.
Are you ready to announce our guest today?
I am. Our guest this afternoon is Dr. Alan Warren. He is a professor of hematology at the University of Cambridge in the United Kingdom. He's also the clinical lead for bone marrow failure and myelodysplastic syndromes at Cambridge University Hospitals. His lab actually discovered that the defective assembly of ribosomes, the machines in all of our cells that make protein, is what causes Shwachman-Diamond Syndrome. It's huge, 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 huge. Dr. Warren's going to explain his new drug therapy research that will stop the progression of SDS, and that the All In campaign is a collaborative effort being sponsored by the Shwachman-Diamond Syndrome Foundation, the Shwachman-Diamond Project, and the SDS UK group, which I think is the first in history to have this big of a collaboration together for such a rare disease. So I would like to go ahead and welcome Dr. Warren.
Hi, Dr. Warren.
Hi. Pleased to be here this evening. It's dark in Cambridge this evening.
Yeah. I know. I guess we should also say thanks, everyone, for accommodating the time zone difference. We obviously wanted to make it the easiest for Dr. Warren to join us, and I don't think he'd want to join us at midnight. So
Dr. Warren, we're going to hand the floor over to you to present.
Okay. Thank you. So thanks for everybody for joining here this evening. So I'm going to try and open this PowerPoint presentation. Can you see? Can you see the slides? So what I'm going to do this evening is to give you some of the background to the project that we've initiated to basically target the ribosome to try to devise a therapy to treat Shwachman-Diamond Syndrome. So I want to give you enough background to try to understand why we might want to do this. Why might we want to try to target the ribosome to treat Shwachman-Diamond Syndrome? So I'm going to summarize some of the work that we've been doing in my lab over the last many years, and hopefully communicate this to you in a format that's oops, my Chrome has now disappeared. Can you see the slide? Yeah. So this is a picture of a HeLa cell. This is a section through a cell, and what I want you to notice, I hope you can see the pointer, are these little dots which lie on the surface of the membrane that encircles the nucleus, and these are ribosomes. So these are ribosomal subunits which are composed of a large and a small subunit. And what's amazing is that every minute, about 20,000 of these ribosomes are exported from the nucleus of the cell into the cytoplasm, where these ribosomes have to complete the process of maturation. So ribosomes are the machines that make all the proteins in the cell, and they're constructed by a process that's somewhat similar to a production line, a car production line, where there's a number of workers, enzymes and factors that all contribute to making these fully functional ribosomes, and there are processes in the cell that interrogate whether these ribosomes are fully formed and functionally competent to make protein. And based on the work that we've done over the last many years, we think that the protein that's predominantly mutated in Shwachman-Diamond syndrome, the SPDS protein, is involved in this quality control interrogation of the maturing ribosomes. So I need to give you a bit of background as to the anatomy of the ribosome itself. So the ribosome is comprised of a small subunit, which is shown on the left, and the large subunit on the right. And most of this spaghetti, if you like, is the RNA. So this is the ribonucleic acid that makes up most of the ribosome. And there are a number of additional proteins which coat the surface of the ribosome. The messenger RNA, which is the genetic code, is read by something called the tRNA, which is an adapter RNA protein. And the decoding of the genetic message is done by the small subunits, and the protein is then assembled by the large subunit, and it's extruded through this channel, which runs all the way from this face of the large subunit that faces the small subunit. It runs all the way through the back of the ribosome and then out into the cytoplasm. So this is called the nascent polypeptide. And this movie summarizes how the ribosome decodes the message. As I said, it involves this adapter molecule, which is the tRNA, and an enzyme which is called Elongation Factor 2. And you can see here in this movie the protein is being assembled, and gradually it's extruded through the exit tunnel and all the way out to the cytoplasm. So as I say, the subunits are, the intact ribosome is composed of a large subunit and a small subunit. And the sort of business end, the catalytic site where all this action is happening, is something called the peptidyl transferase center. And I'll come back and discuss this in a bit more detail later. So the ribosome is a machine which is involved in reading the genetic code and using that information to make all the proteins in the cell. So when Joanna Romans identified the SPDS gene as the as the gene which was predominantly mutated in patients with Shwachman-Diamond syndrome, the question arose: what is the function of this SPDS gene? So how might we go about trying to reveal the function? So the experiment that we initially did, because we like doing structural biology, is we initially crystallize the protein from prokaryotes, so from cells that live in volcanoes and hot springs, because these proteins tend to be highly stable, and we were able to generate a very high resolution crystal structure. So this shows the 3-domain architecture of the archaeal SPDS protein. And on the right is the structure of the human protein. As you can see, they look very similar. And these are all the missense variants, which have been identified, mapped onto the surface of the SPDS protein. And when we initially solved the structure, it became apparent that this domain here, this domain in green, it was similar to another enzyme called Elongation Factor 2. And at the time, when we solved the structure, the suggestion was that maybe the SPDS protein was actually a factor which cooperated with some of the translational machinery. But we had to do some genetic experiments to try to tease out the function. And because the human SPDS protein is so highly conserved from humans to prokaryotes to cells without a nucleus, we were able to take advantage of this and exploit yeast as a genetic tool to try to elucidate the function. So we took yeast cells which were completely lacking the homologs of the SPDS protein in yeast, and the cells grew very slowly. But what we found was that over a period of 2 or 3 days, the yeast cells would recover the ability to grow in liquid culture. And through a long process, we were able to map the protein that we were able to find mutations in this protein called EIF6, which stands for Initiation Factor 6. And we found 24 different amino acids. So all of these variants mapped to one face of this EIF6 protein. And the idea is that these mutations, these variants, affect the ability of this protein to bind to the ribosome. So why is that important? And the reason is apparent from this structure from Sebastian Klinger. So this is the small subunit, the large subunit, and here's EIF6 on the surface. And as you can see, there's a clash. So the EIF6 protein sits right at the junction between the large subunit here on the right and the small subunits on the left. So it forms a barrier to the assembly of these two subunits into an actively translating ribosome. So it needs to be removed in order to allow the assembly of productive ribosomes, things that will make protein efficiently. Can move to the next slide. So again, through the process of yeast genetics, we were able to identify another factor with which this Shwachman protein, SPDS, that it cooperates with functionally to release EIF6 from the ribosome. And this other factor is called Elongation Factor like 1. It's an enzyme which it breaks down something called GTP to work. So this is the enzyme, and SBS is a cofactor for EFL1, and the two proteins together drive release of EIF6 from the ribosome. So the model that we developed with using genetics and a lot of biochemical experiments that I'm not going to go into here, the idea is that the immature large ribosomal subunit joins with the 40S subunit following the release of this factor called EIF6 through the action of SPDS and EFL1. So this following the release of EIF6, the mature 60S joins with the 40S to form this machine, this actively translating ribosome, which then makes all the proteins in the cell. And what goes wrong in Shwachman-Diamond syndrome is that the SPDS, or indeed the EFL1 protein, in some cases is deficient, and as a result, EIF6 gets stuck on the surface of the large ribosomal subunit, and these two subunits can then no longer come together. So there's a joining defect, as it's termed, And this reduces the amount of mature, actively translating ribosomes in the cell, and the cells make less protein, and that's why we see the different clinical phenotypes. It's as a result of the reduction in protein synthesis in the cell. But seeing is believing, and so what we wanted to do next was to try to visualize these proteins on the surface of the large ribosomal subunits. So we wanted to try to develop methods to actually see the SPDS, EFL1, and DIF6 proteins together to try to understand how they all work and do their job. And at this point, we were based in the MRC Laboratory of Molecular Biology in Cambridge, where there was a revolution going on in a technique which is called single particle cryoelectron microscopy. So this is a method that allows you to actually see the individual side chains, and if the resolution is high enough, we're able to reconstruct images of the entire ribosome with all the factors bound, so we can see the sort of nuts and bolts in incredible detail. And this was due to developments in mainly the detectors that were used and software for deconvoluting all these the images of the ribosomes on a grid. So by applying this methodology, we were able to visualize SPDS, EFL1, and EIF6 on the ribosome. So first of all, this is the SBDS protein. So what you can see here in cyan and blue is the large ribosomal subunit. And this is the face of the large subunit that binds to the small subunit, or the 40S. And here you can see the SPDS protein, which is actually binding in the key functional site of the ribosome, which I mentioned earlier, the so-called PTC, the peptidyl transferase center, which is where the proteins are stitched together. This movie again shows the peptide, which is coming out through the exit tunnel. So the SPDS protein is actually plugging the exit tunnel so that protein is not able to pass through this tunnel until the ribosome is fully mature. But as you can see, there's no direct contact between the Shwachman protein and EIF6, which is blocking ribosome assembly. So the question is, how does SPDS actually facilitate the release of EIF6? So we're lucky to be able to build models of both SPDS and EFL1, so that's what I'm going to show you next. So here is the movie, if you like, of SPDS, which is shown here in magenta, and here's the EFL1 protein in blue, and EIF6 is in yellow. I'm just going to swing this round to the side, and so again you can see there's no direct contact between the SPDS protein and EIF6, but the enzyme EFL1 is directly binding to EIF6. And then we were able to collect additional particles which were completely lacking EIF6, and as a result we put together this movie of how we think these factors cooperate with each other to drive release of EIF6 from the ribosome. So EIF6 is shown here in yellow. So what you see is this large enzyme which is recruited to the ribosome by binding to the Shwachman protein, and then it changes shape. There's a sort of change in the relative orientation of the different domains of this protein, and it competes, the EFL1 enzyme competes with the EIF6 for the same binding site on the ribosome, and this results in this protein being displaced from the surface. So, this is really astonishing at the time that we're able to actually visualize all these factors bound to the ribosome and propose a model which has been supported by or of biochemistry and genetics as to how these factors work together. So the key defect, the key problem in Shwachman-Diamond syndrome is the inability to efficiently remove EIF6 from the surface of the large ribosomal subunit. So, as we went forwards, it became clear that SPDS is the predominant protein that's mutated in this disorder, but there are others. And in fact, multiple components of the ribosome assembly pathway are also mutated in human disease. And so we wanted to try to understand the entire the entire assembly line, if you like. So how is the large subunit actually put together when the pre-ribosomes emerge from the nucleus into the cytoplasm? Because we wanted to build the tools to try and understand this pathway in really high amount of detail, so that we could potentially think about ways of targeting this process to treat these diseases. The first, so Stepensky and reported the mutations in EFL1, which also the patients also have a Shwachman-Diamond syndrome phenotype. And we, together with Patrick Revian, we also identified 3 families with mutations in EFL1. As you can see here, these are ribosome profiles. Essentially what they show is that there's an assembly defect, and down here we measured the ability of the cells to make protein, and in the absence of the EFL1 protein, the cells are quite sick, so they make less than 50% of the wild type amounts of protein, so they're quite severely impaired. And then work from Yigal Dror and Indrajit Dokal showed that a subset of SDS patients carry mutations in this other factor, which is called DNA JC21. And more recently, 2 groups, so Jean Donager and Seamak Bayram identified mutations in this SRP54, which is machinery that allows the ribosome to load onto membrane and transfer the protein into what's called the endoplasmic reticulum, which is the defect that we see in pancreatic cells in Shwachman-Diamond syndrome. The protein is poorly able to move into the ER and be secreted from the cell. And then Kim Kiersmacher has shown mutations in this factor. It's a ribosomal protein called UL16, and mutations in UL16 are found in patients with a subtype of leukemia called T-cell acute lymphoblastic leukemia. So the point is that many different components of this ribosome assembly pathway are targeted in disease, and so we wanted to try to visualize how all the ribosome assembly factors come together to produce a mature ribosome, and to try to order this pathway, because the order of these assembly events or these maturation events was not clear when we started out on this journey. So I'm going to show you a movie as to how the ribosome undergoes the final maturation steps. And the key thing that we wanted to understand by doing this, and again we're using the Cryo-EM technology, using these fancy microscopes, we wanted to understand when is EIF6 released, so when does SPDS and EFL1, when do they do their job, because there was quite a lot of debate as to the exact timing of release of EIF6. So all of this, the spaghetti here is the RNA, so this is the ribosomal RNA, and these coloured objects are the assembly factors, which all lie on this surface of the large ribosomal subunit. And what needs to happen is that all these factors need to be released to open up this space on the large ribosomal subunit to allow it to join with the small or 40S ribosomal subunit. So what I'm going to show you is there's going to be a lot of changes in the positions of these pieces of RNA. Many of the assembly factors are going to be dissociated, and there'll be recruitment of some of the final ribosomal proteins. So I'll just hopefully set this running. So the details don't matter. It's just to give you an idea of how dynamic this entire process is. So this is one of the first proteins to go, ARX1, and then RAI1 is dissociated from this exit tunnel, and it's replaced with another factor which plugs the protein exit tunnel until the particles fully mature. If we swing back round, so this factor is released, and we have recruitment of one of the ribosomal proteins, which is called L40. So this protein docks at the so-called P stalk, and then we recruit this UL16, which, as I said, is mutated in T-cell acute lymphoblastic leukemia. And as a result of this piece of RNA now undergoes a dramatic change in position. It swings out of the way, and then this destabilizes this huge protein in red here, which is called NMD3. And then some magic happens, and these two factors are dissociated from the surface. And now what happens is the Shwachman protein comes in to interrogate whether the ribosome is mature. So it comes and interrogates the loading of this UL16 protein. Here comes EFL1, and then the two proteins cooperate with each other to then drive release of EIF6. So we think this is the final step in the maturation of the large ribosomal subunit. And SPDS and EF1 are acting as what we call quality control factors. So these are the last factors in this production line, which make sure that everything's in place, all the RNA is in the right position, and we've had incorporation of all the ribosomal proteins, including this UL16 protein, which runs right down into the key functional site of the ribosome. So this subunit is now good to go, and is now able to enter into active translation by joining with the small 40S subunit. So again, just coming back to the model, we think that the release of EIF6 is the final step in the maturation of the large subunit. We're still trying to work out the details of this process, and there are some additional factors involved, but essentially what I've shown you is conserved to a large extent from human to prokaryotes. Or cells without a nucleus, and actually the SVDS protein, from humans to prokaryotes, cooperates with an enzyme, EFL1 or an EFL1-like protein to release EIF6, generate a mature 60S, which then joins with the mature 40 and forms these actively translating ribosomes. So I want to move on and just discuss this question of how do stem cells in Shwachman-Diamond syndrome, how do they escape the ribosome assembly defect? You might expect that because of the defect in the ability to make protein, that the stem and progenitor cells in patients would have what we might call a fitness defect. And this has become known as Damashek's riddle. So as I told you already, initially back in 2007, we'd identified all these mutations or variants in EIF6, which map to the surface of EIF6, which binds to the ribosome. And so these mutations allow the cells to completely bypass the absence of the Shwachman protein by reducing the ability of this protein to bind to the ribosome. So we asked this simple question: does this happen in humans as well, in patients with SDS? And this was a really nice collaboration again with Patrick Reevy, where indeed we identified a whole variety of variants in the EIF6 protein and gene, and indeed they're frequent. These are frequent mutations are frequent in patients with SDS. The mutations are all shown here. There's a number of recurrent mutations, particularly this mutation called N106S. But there's a whole range of missense mutations and other mutations that are these nonsense mutations, which should, in theory, give you no functional protein at all. Elena Mazzarati and actually Yigal had noted years ago that patients with SDS had these interstitial deletions on chromosome 20. This is one of the most common chromosomal changes in SDS, and what happens is that the EIF6 gene is lost as a result of this deletion. And it was always difficult to pin this down on EIF6 specifically, because there were a number of other genes which are lost as a result of this deletion. But we found this reciprocal translocation, which specifically affects the EIF6 gene. So again, suggesting that it is indeed the EIF6 gene which is important in this deleted region. So the next thing, having identified all these mutations, and I should say this was also found in a lovely paper from Akiko and Coleman Lindsley, we then set out to test the functional consequences of these mutations. So Alex Fine in the lab has again used this technique of cryo-electron microscopy to generate a very high resolution structure of the human EIF6 bound to human ribosomes. So we can see at a very detailed level the interaction between the EIF6 and the human ribosome. What we found was that there are the variants mapped to 3 distinct sites, so a site away from the surface of the ribosome, and most of these variants actually unfold the protein. Then we had variants that map to the interaction with the ribosome, to this protein here, and a separate set of interactions to, again, to this, one of the other ribosomal proteins, this time L24. Okay. So we did set out to use this as structural information to interrogate the consequences of these of mutating these variants. And I'll just summarize this experiment here. So what we're doing here is to add the wild type EIF6 protein, and this is enough to cause a ribosomal subunit joining defect, because the EIF6 binds to the surface of the large subunit and prevents the 2 subunits coming together. So what we see is the accumulation of the small and the large subunit, and a bit of a reduction in the number of fully mature ATS particles. Whereas if we added the EIF6 carrying one of these variants, that was not the case. So in other words, the EIF6 protein has a reduced capacity to bind to the ribosome as a result of this N106S variant. And this was one of the variants actually which we'd found, some of the variants we found years ago in our yeast experiments, and it was recapitulated in the SDS patient cells. If we take yeast cells which are lacking SPDS, we can show that they have a growth defect. So at 37 degrees, these are SPDS deficient cells which fail to grow efficiently. But if we add this variant, we can restore the ability of the cells to grow. So these EIF6 variants, they either completely reduce the expression of the protein, or they reduce the ability of EIF6 to bind to the ribosome. And just for fun, I'm going to show you some, these are called molecular dynamic simulations, and it's just to show how the N106S mutation actually disrupts the interaction between EIF6 and the ribosome. So on the left is the wild type protein, and you can see that these are the side chains of the amino acids which are held. There's a very tight interaction here. These are little water molecules running around on the surface. Whereas if you look at the mutant, so this is the N106, this is the N106 here, this is the serine 106. you can see these water molecules become lodged at the interface between EIF6 and the ribosome. And what this is telling us is that the effect of this N106S mutation is to completely destabilize the interaction between EIF6 and the ribosome. These experiments are run over a few nanoseconds, so thousandth of 10 to the minus 9 nanosecond. And if we run it for longer, what happens is the EIF6 then eventually falls off because the interface becomes completely replaced with water. So the next experiment we want to do is to ask whether if we introduced one of these mutations, these variants into a fly, which was lacking the SPDS protein, can we restore the viability of this Shwachman fly model? So what you can see here is the wild type flies, and these are larvae, which grow into adults, whereas they're SBDS deficient flies, and they're poorly viable, and they die around what's called the third instar stage, so very early in development. And if we express this variant, this N106S, in our SPDS mutant flies, what was really astonishing is that we can completely rescue the viability of this whole animal. So these animals are viable and fertile. And also, if we simply deplete EIF6, if we knock down the amount of EIF6 in the fly, it wasn't quite as efficient as this variant, but we're able to, again, at least partially rescue the development in this whole animal model. So this is the kind of background that led us to think, might it be possible to mimic the effect of these EIF6 variants in order to produce a treatment, a drug therapy to treat patients with Shwachman-Diamond Syndrome. So can we mimic the effect of this N106S variant? So just to summarize this work, so what we've described in these SDS patients is the concept of somatic genetic rescue. So somatic cells are able to rescue the underlying germline defect in ribosome assembly, either by reducing the dose of EIF6 or by reducing the ability of EIF6 to bind to the ribosome as a result of these missense variants. So is precision medicine possible for SDS by trying to design so-called EIF6 suppressor mimics? And I just want to show you a video which was recorded back in 2019 when the BBC came to the lab to visit, just to give you an idea of what we're trying to do here. So I'll just run this video.
The final 3D image of the ribosome, complete with troublesome yellow lump, a protein that's meant to break off but gets stuck, causing a rare but dangerous condition called Shwachman-Diamond Syndrome.
It's exciting we have these new technologies, the new microscope facility.
In his Cambridge lab, Professor Alan Warren is using the pictures to find a drug treatment. Like testing keys for a lock, it's about matching up the shapes.
So you literally need to zoom into here and see the shape that you're trying to find. You're looking for a drug to fit that shape effectively.
Correct. So the idea is that we could find a drug which might sit at the interface between this protein and the rest of the ribosome. So we want to find something that will get into the groove and knock off this protein from the surface of the ribosome. So we're trying to get in the groove,
the final,
as it were. So I'm not going to give you the nuts and bolts of how we've gone about doing this to date, but I want to show you something that got us very excited, because this was just one of the initial hits that came out of some of the screens that we've been doing, and we've tested this hit, if you like, in a screen to see whether we can rescue the viability of our Shwachman fly model. So, as I said, the SDS mutant flies die around the third instar larval stage, but by adding, so we wanted to ask whether by adding some of these molecules, can we rescue the development of the fly? And here we can see that compared to the control, or just the vehicle, which is the solvent that we use to dissolve the compound, we can see a considerable, or we can see a rescue of the proportion of flies that develop to the pupal stage. And so this was very exciting to us, and in fact, we've now been able to, as a result of experiments like this, we have had significant investment from venture capitalists, Medici, to try to facilitate taking this project forwards and try to develop small molecule EIF6 suppressor mimics that might be useful as a potential therapy in the future for SDS. I think, I just wanted to say that we've also developed a new mouse model, which is different to the ones which are currently being developed. The more mouse models that we have, the better. Of course, this mouse model is still viable, but it's an EIF6 overexpressing animal, and we can regulate the amount of EIF6 by inducing this so-called TEC transactivator. So we add a molecule called doxycycline, and we can induce a graded increase in the overexpression of EIF6. If we overexpress too much of it, it's lethal, but we can dial down the dose of EIF6. And the animals are still viable, but they just have a mild anemia. So we think that this might be useful in terms of screening the small molecules that hopefully are going to emerge from this program of work. And this was developed by Pekka Yeko, and the publication is currently in bioRxiv. Overexpressing EIF6, it mimics the Shwachman defect because it induces a ribosomal subunit joining defect. And the idea would be that a small molecule that interacts with EIF6, we can test whether we can inhibit the activity of EIF6 and thereby restore ribosome homeostasis. So I'm going to finish there and just thank all of you for your attention. These are all the people in the lab who've been helping us over the years. We had support from the Medical Research Council, from and Blood Cancer UK. So Blood Cancer UK initially got us going on STS following. I have to acknowledge Kim Wright, who I spoke to very many years ago, who got me interested in trying to work on this condition in the first place. I'd like to thank Ted's gang on the Conor Wright Shwachman-Diamond project, who've been very supportive over the years, all the STS patients and their families. And I'd like to thank the SDS Foundation, SDS UK, and the Shwachman-Diamond project for their support to try and take this project forward. So I'll finish there and take your questions.
Very interesting, Dr. Warren. That was, I was on the edge of my seat just watching it, and we've talked about it a lot over the last week. So you'd think I would know, but it was so interested. I was so interested. We had a couple of questions come in. Nicole, do you want to head up? Do you want to start?
Sure. From Lisa Saporina, can you please explain your phases of development and estimated timeline for phases?
So it's quite early to start talking about the phases. We're still at the very early stages of this project. It's going to take us a few years probably just to get some lead compounds. Obviously, because it would be a new molecule, we need to go through phase 1, which the initial test would be in adults for toxicity, and then so a sort of single ascending dose type schedule, and then multiple ascending dosing in adults with Shwachman-Diamond syndrome. So I'm being quite cautious as to how long this is going to take. I don't want to give false, optimism, but it's going to take a while.
Yeah. Next question is from Justin Grinnell. Can you explain how this research will mean for clinical families?
So if there are, as I said, so patients with mutations in SBDS or EFL1, we would expect that a drug targeting EIF6 would help to rescue those patients. So any other patients with a mutation which is currently unknown, but it's possible there are other patients where the mutation is affecting another component of the assembly pathway. But the sort of common downstream consequence of that is likely to be failure to release EIF6 from the ribosome. So we would hope that such a molecule might be able to help all the families.
The next question we had come in was, if multiple components of the ribosome assembly are targeted, what could this mean for the future of SDS as a whole body disorder?
So what we're trying to do with such a therapy, the concept is that we could restore ribosome homeostasis, so the balance of protein synthesis in all cells. So does that answer the question? So we'd hope that we could target the blood system, but also targeting cells beyond blood, hopefully.
No, I think it answers it really well because, like you said, the ribosome is so much more than just blood. It's so much more than that, and so it has a lot of impact in the body. The next question we had was from Katie. Are you personally involved with the mouse project from SDS Alliance? And could you explain the significance of the project and its relations to the work that you're doing?
So yeah, I mean, I've had conversations about this mouse, and I think the more mouse models that we have, the better for the community, because it gives us much, each mouse model has lots of value for, a small molecule is not the only approach to therapy. We might think about gene therapies or stop codon read-through or affecting the splice mutation, for example. So I think we need a diversity of approaches. Our own involvement with this was to, once the mice have been generated, to get involved in looking at the ribosome function in these animals and characterize protein synthesis. And potentially it could be a very useful tool for us and for the whole community, actually, in terms of thinking about how to treat SDS.
Yeah, the next question we had come in was from Joan. Has this approach been tried with other diseases?
So making small molecules. So I think cystic fibrosis is a good example where over the last, I guess it's been 30 years in the making. I think Joanna spoke about this at the last meeting. So there are now molecules which target the CFTR protein in different ways, and these are now in clinical use. So yes, small molecule therapies for disease there is historical precedent for them. It's very exciting.
Yeah, it sounds, I know it's very exciting for all of us here. The next question we had to come in, which is actually kind of really interesting and fun, is from Dr. Corey. He said, very nice work, Alan. One question is whether therapeutic intervention is necessary during fetal development or whether postnatal is sufficient to mitigate multiple organ phenotypes. There's a lot of big words in there, so I'm hoping you can unpack them for the rest of us little people down here.
So yeah, thanks, Seth. Nice to see you. Your picture, at least. So yeah, I guess the earlier you would start such a therapy, the better. I'm not sure. Yeah, we'd have to start with clinical trials and see how it goes. Can we get it? Do we have efficacy in adults and children, and then try to work back? But I guess the sooner a therapy was started, the better.
Absolutely.
Really exciting.
I've loved if we scroll back up, we can see the Shwachman-Diamond Syndrome registry is here. I believe there were a couple from SDS UK that jumped on. I know there's multiple people watching from YouTube. This is just such an exciting research project opportunity, and we feel like it's really going to be the future for SDS. It's just, we're all very excited about this.
Dr. Corey just asked, can we get SPDS incorporated into newborn screening? Ooh.
Oh, Dr. Corey with the hard hitters.
This is why it helps to have doctors in the comment section. They ask the good questions. Not that everyone's questions isn't good, but they ask the really hard questions.
Yeah, that would be great.
How do we do that, Dr. Corey? Do you have a 12-step plan for us to get there?
Probably not.
Probably not. He's like, no, I was hoping Dr. Warren had the 12-step answer. That's why I asked. Dr. Warren was supposed to have the answer. Go ahead, Nicole.
Hello from the UK. My son, 3 years old, has SDS. What will that mean for him, for the future for him? That's a good question for a 3-year-old. What could this mean for a 3-year-old right now?
Yeah. So, I guess it needs to be, has he been referred to one of the main centres for expert care? So that would be the key thing to start with. Where are you? I can make contact with you and make sure that you are being, followed up in the optimal manner. So you need to access multidisciplinary care in a large hospital centre with expertise in managing SDS. If you contact me, I can point you in the right direction if you haven't already.
If you message us afterwards, we have the contact information for Dr. Warren and how to get involved with anything or get a hold of them, as well as for the registry. We have the registry information. We have Dr. Warren's information. We really can get you in touch with. That's, I think, my favorite part about these shows, Nicole, is that we actually bring doctors into people's living rooms, and you're able to feel like you connect with them. Yeah.
Good.
Dr. Corey pawned it off. He said, can the SDSF do it through the states? I don't know that answer. I don't have that answer. I'm going to let Joan maybe take that answer, and that's something we could look into, because I don't even know how to get something on a newborn screening. If there's like a panel of doctors somewhere that make big decisions, I don't know. I have nothing on that, but that'd be great. Dr. Warren, I think we just want to say a collective thank you from the entire SDS community for putting your heart and soul into this project, as well as all of your other ventures, growing roses, learning new languages in your spare time. For a community that's not heard very much, we feel very heard by the doctors who fully stand behind us. And we know this research is going to be huge, and it's going to be awesome. One last question that just came in from Paula. Do you feel that this treatment could be available to our kids in their lifetime?
So yeah, I would hope so. That's the plan. Yeah. So yeah, we have good intentions to try to do this as fast as we can, with the support from these venture capital community. Then hopefully that'll expedite progress. But yeah, I can't make any guarantees, but we'll work as hard as we can to try to bring this to clinical trial.
Dr. Warren, we are so hopeful, and want to thank all the researchers and doctors that have been working on SDS for decades, and I know all their work feeds into this work.
I have one last question that just came in. Sorry, it was via Messenger. That's why I picked up my phone. I heard it ding. Sarah had asked, what was administered to the fly/mouse, and what type of drug do you anticipate that will come from this? An injection, a pill, infusion?
Oh yeah, we forgot to ask.
So yes, these were some of the initial hits that came out from the screens. These were just the first initial compounds that haven't been optimized in any way at all. So it was added into the fly food, and remarkably, we found that it was able to rescue the development of these flies. So this compound needs to be obviously modified and made more efficient. And there's a lot of medicinal chemistry that now needs to go into trying to try and develop this as a drug candidate potentially. And then, I guess ideally we'd want to administer a drug orally, but it's a long task ahead to get it into that state. So.
Absolutely. Multiple comments coming in. Thank you from Connor Wright's family watching from the UK. I'm hopeful that you know who that is because I'm not from the UK. Just lots of thank yous coming in across the board. I hope you can see them and feel that this will be such a huge, huge project for families and everything that you do for us.
We want to thank Greg as well. He runs the behind the scenes tech on the show. We've never worked so hard on this show. Michelle and I have 3 screens going, and our phones have multiple avenues of communication going with questions coming from our foundation page. There are lots of different questions coming in. So thank you for being patient with all the questions coming in.
Any questions we didn't get to, we will write them down, take them, and email them accordingly to where they need to go to. Probably a lot to Dr. Warren, I would hope, after this one. But as always, we are more than willing. Just because your question did not get asked on the live, it does not mean it was dismissed. I want you to know we're working very hard, even off screen, to get you your answers.
Thank you, everyone. Thank you, Dr. Warren. Thank you so much for your time. And thank you, everyone that joined us in the comments.
Yes, thank you, Dr. Warren. A wealth of information today, for sure.
If I want to remind everyone about the giveaway today, just like, comment, share, start a watch party. Even if you're logging on late and watching this off of YouTube or Facebook late, go ahead, comment, and you're still in the drawing. If you haven't already joined the SDS registry, it is so important. I know we have a lot of people, we're talking about the US registry, but I know other groups have their own registries. It is so important, the US registry. We want to specify US registry because we have lots of different groups joining us today. Thank you so much. And we're stronger together. So that's why it's so important to join that. And I think we got all the questions, right, Michelle? I mean, there's a good chunk. There's a couple we missed, I'm sure, because they're coming in so fast, but we can try and figure it out and how to get there. Derek Graham, can we pull Dr. Warren back up? Oh, yeah. Is Dr. Warren still here? Or did he go? Oh, there he is. Last question, and we promise we'll let you go. Sorry. Derek Graham, my son is an adult with SDS. How can we sign him up for future clinical trials?
So just stay in touch. Send me a letter. We'll get your details, and we'll keep you up to date as this project moves forward.
Okay, thank you. Yes, you can watch for updates probably from SDSF or Shwachman-Diamond Project or SDS UK. We'll all be posting updates on this very beneficial research. So when we know, you'll know. Thank you, everybody. Thank you, Dr. Warren, for your time. Thank you, everyone in the comments. Yes. Have a wonderful night. Yes. Good night, everyone. Good day. Good day. Good afternoon. Good day. Good night. Good evening. Whatever time zone you're in, have a good one. Thank you. Thank you, everybody. Wash your hands. Wash your hands.